Yes, so my talk will be from completely different universe that the previous session was about. So I thought I would start, and recognizing that from the program, I thought I would start with the basic introduction about what we study. So that so my talk will be about modeling biological macromolecules, such as proteins that fill biological cells, and also build viruses, and where they perform all kinds of functions, including, for instance, enzymatic reactions. Proteins and other molecules usually do not act alone. They interact with each other to form larger assemblies, which we call complexes. And those complexes usually adopt a defined function, defined structure that then determines their function. And protein structure and structure of complexes is quite complicated. So we always have this polypeptide chain that falls into so-called secondary structure of beta strands and alpha helices, which then interact with each other to form a tertiary structure or a structure of a single protein. And those tertiary structures interact with each other to form quaternary structure or a complex. And why it is useful to study the structure? For example, the structure allows understanding, helps understanding how proteins work, how they perform the structure. For example, this movie is an interpolation between multiple experimental structures of ATP synthase, the enzyme that synthesizes ATP. And this video on the structures actually explain how ATP is synthesized in cells. This structure depicts a drug molecule bound to a structure of the main protease of coronavirus, and indeed structures can be used to understand how drug works and also to design new drugs. And here is the cryo-electron microscopy structure of a beta coronavirus published before the pandemic. That was still before the pandemic, used to design a mutant that stabilized the spike protein in a pre-fusion conformation. And this structure was crucial to then design the vaccines that ended the pandemic. How are structures determined? Again, it's a bit basics for those who are outside of the field. So there are several techniques. We have nuclear magnetic resonance, X-ray crystallography, cryogenic electron microscopy, and cryogenic electron tomography, cryo-EM and cryo-ET. And these methods often allow to determine the structures at very high precision. For instance, cryo-EM and cryo-ET and X-ray crystallography deliver density maps, electron or optical density maps, that are precise enough to build atoms directly in those maps and derive the atomic structure. But often, especially in the case of cryo-EM and cryo-ET, these density maps are of low resolution. For instance, here there's a map of a complex at around 20 angstrom resolution. So at 20 angstrom, you cannot build atoms directly to solve the structure. And this map represents a human nuclear pore complex, which will be also the main example for use in my talk. The human nuclear pore complexes are proteinaceous channels that are embedded in the nuclear envelope that surrounds the nucleus of every eukaryotic cell, or almost every eukaryotic cell. And they are responsible for transporting macromolecules between the nucleus and the cytoplasm. They are huge. They are one of the biggest complexes in the eukaryotic cell. They are composed of 30 different proteins in many copies, accounting for 1,000 proteins, and leading to 120 megadalton of molecular weight and 120 nanometer in diameter. Those complexes can be visualized with cryo-electron tomography. Here is a cryo-electron tomogram of a nuclear envelope, and in the red circles I indicate those nuclear pore complexes. You can see that's a very low resolution image. Definitely you cannot see single proteins, but what we can do in cryo-electron tomography of biological samples is that we can pick many of instances of the same complex and then align together an average to get a higher resolution map. Like this one. So this map that I mentioned yesterday, just before, is the average map coming from many tomograms like I just shown. Could X-ray crystallography solve the nuclear pore complex? Not really. So far, no one managed to even purify the human nuclear pore complex in an isolated form, and also no one, of course, managed then to crystallize the complex. For human nuclear complex, also the high-resolution single electron microscopy was not yet successful. But X-ray crystallography has been used to solve individual proteins or domains of parts of the nuclear pore complex or subcomplexes, especially for human, but also other species. Some structures could be also actually predicted. So in biology, we can also actually model protein structures. And until recently, the main method was called homology modeling. And this will be important for the rest of the talk when I explain the revolution that we undergo. And in homology modeling, for instance, if you want to have model have a structure of a human protein, but we cannot solve it, but we have a structure of a similar protein from another species, we can actually, using very established methods decades ago, we can combine this information to model the sequence using the other structure as a template to get a homology model of the human proteins. These models were often with quite limited accuracy, but were super useful. So you can imagine that once we have structures of the pieces of the complex and this low-resolution electron microscopy map, we can actually, like a jigsaw puzzle, put the structures into the map. So every blob in this grayscale map corresponds to one of those proteins I show on the left. But at this resolution, it's often actually non-trivial to find an ambiguous placement of those structures in the map. So that's why we often look for orthogonal information. And one of those orthogonal information comes from something what you can imagine as molecular rulers. So we can use special compounds called crosslinkers that would covalently connect different specific residue pairs or residue types in a protein or protein complex. This technique is called cross-link mass spectrometry. I'm skipping the details how it works, but the output is that for every sequence in the complex, which are those horizontal bars, you can get some connectivity to the other parts of other proteins. So these lines that connect the different bars are those crosslinks that inform that these residues have to be close together in the complex because they got connected with this crosslinker. So now having those pieces of the puzzle and the restraints, which ones should go together, we can fit them to the map. And of course, we don't do it manually. We use computation, which we call integrative structural modeling, because we integrate different experimental restraints to build a model. And this is what we did for the nuclear pore complex in 2016, when I was still a postdoc in Heidelberg, that we took this map that I just showed you at 23 angstrom resolution, crystal structures and homology models I built for missing proteins, and distance restraints from cross-linking mass spectrometry to model the human nuclear pore complex. And to model that complex, I started developing at the time my own modeling pipeline, which I called assembline, because it's an assembly line of macromolecular complexes, which we finished and published recently, and which is a pipeline based on a modeling library called Integrative Modeling Platform developed by Andrew Shally at UCSF. How it more or less works, so it usually starts with electron microscopy map like this one. And then we take crystal structures or homology models of individual proteins or subcomplexes. We coarse-grain them to some bit representation. And then we use some special procedure that very quickly and efficiently finds possible combinations of these components in this map, taking the cross-correlation with the map and those cross-link restraints into account in the same time. And this is a Monte Carlo procedure that stacks in the local minimum very quickly. So usually you would run many simulations in parallel, starting from different orientations and fitting them in different order. In the end, depending on the size of the complex, ending with many models, thousands or hundreds of thousands. And then these models are often very clashy and very imperfect because we coarse-grain so much. So then what we would take, we would sort the models, take the top models, as many as our computer cluster is big, and then we would decrease the coarse-graining and refine them in the map in a more continuous space to provide more detailed models. Sometimes still in a bit representation like here, or even atomic representation. And then we perform some analysis, compare the models, cluster, assess confidence. Sometimes we iterate to repeat everything if the models don't converge. And then at the end, we back project them to the atomic representation. And just to provide some more technical details, assembly uses coarse-grained multi-scale representation, so it can use low-resolution bits and high-resolution bits in the same time. It uses a standard classical Metropolis Markov chain Monte Carlo optimization algorithm, linear function for as a scoring function as energy for optimization. And the key is some special multi-step procedure to narrow down the conformational space that even if we have 10 or 20 rigid bodies, that it still somehow works. So there are some heuristics there. So this assembly and this approach I used in 2016 to model the human nuclear pore complex, but actually we haven't finished the job. So that complex only comprised, that model only comprised 30% of the entire complex. And since then I moved to Hamburg. We actually continued the collaboration with Martin Beck to improve the model and maybe complete the missing pieces. So our collaborators have tried to improve the map, and by, I think, 2020, they managed to improve the resolution to 12 angstroms, so still relatively low. And we've been attempting the model. So Agnieszka, the shared postdoc between our lab, was taking this new map and then repeating, painstakingly taking all crystal structure, extra structures that have been solved by then, and improving the homology modeling, trying to fill up the missing pieces to somehow fill all the holes in the model. And then again, using assembly to assemble the model. And in spring 2021, we are getting a model that comprised 35 megadalton, 69% of the scaffold of the structure part, still kind of 35% of the pore, more or less. And when we started writing a paper to publish this updated model, and then this happened. So I don't know if everyone knows and heard about AlphaFold. So in 21, when we've been drafting the paper, DeepMind, which is a subsidiary of Alphabet, connected to Google, published a modeling program called AlphaFold. You maybe heard some headlines. So there were lots of hype and news. It was called the Method of the Year of the year 21. There have been several prizes awarded to the authors. Everyone was excited. It has been justified, so the method has been tested before release already through a competition. So in a structure prediction in biology, actually every 2 years we have a competition of modeling. So in this competition, the organizers would contact experimentalists who are just about to solve a crystal or cryo-EM structure and tell them to withhold the structures, do not release, and give them just the protein sequence. And then the teams from all over the world would try to predict those structures without that information, without knowing the structure. And on this graph, you see the teams that participated in the competition. And the higher the bar is, the better the performance of the team or the program that the team brought was. And you can see that this AlphaFold from DeepMind outperforms everyone by a huge margin. How good the models are? So this is some comparison from AlphaFold paper that was later published, which with the green structure being the experimental structure and the blue structure being the model. And for many cases, they were virtually indistinguishable from experimental structures. AlphaFold also provides confidence metrics, so we can actually assess whether you trust the model or not. It's also coming from this the same machine network architecture, so you can get global scores like those PLDT or PTM score, whatever it is. Also local scores per residue mapped as this gradient on the structure, and also this matrix shows depicts the relative confidence in orientation of different subdomains of the same protein. What is AlphaFold? So AlphaFold is a machine learning-based model for predicting the 3D structure of proteins using only sequence. And this has been trained on previously published experimental structures collected in our database, which is called Protein Databank, as well as using large databases of just protein sequences, even if structure was not known. And although it's difficult to really explain how the neural architecture, what kind of information it uses, it looks for patterns. And one of the patterns that probably the network uses, although it's still a hypothesis that we bring while analyzing its output and architecture, is using evolutionary information. So let's imagine that we want to model the protein of human protein with that sequence, which each of these objects is an amino acid. So AlphaFold would first look for sequences of similar proteins, but from other species, using an approach that also has been developed previously before AlphaFold. And then it would look for patterns. So for instance, here there's one pattern that whenever this red amino acid changes the shape to this green amino acid, there will be another residue in another place that would change to a complementary shape. And this actually often happens in proteins because if these amino acids interact with each other, and if one changes to the big amino acid, the other has to change to a small amino acid. So you can detect this correlation which results from coevolution. And this principle can be used to to predict which residues in the sequence would contact with each other. And there have been many methods before that were trying to do that. Just AlphaFold brought it to a completely different level and probably is also looking for other patterns. How does AlphaFold work? It's complicated. I'm a biologist or computational biologist, but still some parts of this architecture are beyond my understanding at the moment. But and even Demis Hassabis, the CEO of the company, said that by then it has been the most complex piece of AI they developed. More or less how it works. So it starts with a sequence, and then on one hand, on the top, it would look for similar sequences to look for those patterns I just told you, and on the bottom. And in the bottom track, it would optionally look for similar structures already, for similar proteins that already have a structure known in a database, although that is optional. And then it would go through with these two representations, with a sequence representation and this sequence alignment representation, and the pair representation that is holding the placeholder for the contacts between all residues, all against all. And then it would put these representations to so-called evoformer module, which is a transformer or attention-based architecture that is using some language model principles of, for instance, masking of some residues and trying to repredict them, attention to derive these long-range contacts between long sequences, which you can treat as a long sentence. And mix this representation together. So the sequence representation would talk to the pair representation. And then after that, it would move these representations to so-called structure module, which is basically performing a minimization of the structure of already a geometry into the information coming from those representations. After that, it would produce a model and the confidence estimates, and then it would actually go back and recycle. And these recycles are actually important for the architecture. So when it was released, now coming back to the nuclear power complex story, we looked to the so actually before the software was released by DeepMind, the first thing that was in collaboration with EMBL, they released a database of plenty of models. So like, for instance, all models for humans. So we went to this database, and we are surprised to see that all nuclear powering models are there. They can be downloaded, colored by this, together with the scores. And then we started analyzing them. So, and at the moment, it was quite we were worried of, was some suspicious that maybe the software is overtrained and so on, how it would generalize to new proteins. But we already had some other unpublished X-ray structures that were for sure not in the training data set. And we saw that actually the models of structures that had no similar structures in the PDB are spot on. We could validate them by fitting to the electron microscopy map that we had. So probably cannot see much, but these are different models fitted to different locations of the map with significant scores. And for instance, I can just point out this one, which is a super long protein that we always had trouble with modeling. So on the left, you have a homology model that we had before, fitted to this density. You can see that kind of fits, but things stick out and a big part is missing. If you take AlphaFold model of that protein, it fits way better, and also this missing part is there, and it just fit. These were models of monomers. So at that time, AlphaFold could only model single proteins. It couldn't model those complexes. But then, as a scientist from David Baker's lab, which also develops protein structure prediction and design programs, tweeted that if you modify these lines of the code, you can transform the entire program to model complexes. And it was just shifting the residue index. Basically, you could take two protein sequences, concatenate, and just just add 200 to the index, and then suddenly it was tricked to treat these two sequences as different proteins. And she just tweeted that, but then others, especially Sergei Ovchinnikov and Martin Steinegger, took this idea and implemented that in the meantime, the code of AlphaFold was released, of course. That's how she could modify the code. And Sergei and Martin Steinegger created the CollabFold, which is a web notebook, Jupyter notebook interface to AlphaFold, and they provided this feature with this hack. So what we did, we downloaded the Jupyter notebook, converted it back to the script, and then ran on our small computer cluster, definitely not a Fugaku computer, but we had some 100 GPU cards, and some of them we could use over a month, and then started modeling subcomplexes. So it couldn't model the entire nuclear complex because it's too big. The memory requirement of AlphaFold is huge, so we wouldn't fit the nuclear complex to even our high-end GPU. And the question would be whether it would ever optimize. So based on our knowledge and all kinds of experiments, many other scientists all across the world did, like biochemical experiments, which proteins interact just from gels. We are creating different combinations of proteins, 2 proteins, 3 proteins, their fragments, and found that we are getting models that were looking good, and they had good scores. So the more blue, if you see more blue there, that means good predicted quality. And we just went on, and that time was a good time because most biologists did not use GPUs, so we had, I think, 20 NVIDIA A100s on our cluster, and we could use that for ourselves. Now things changed. And then we took these complexes and also compared to unpublished crystal structures that we obtained from Andrew Hertz at Caltech. And we saw that, okay, we could actually predict complexes of proteins with short stretches of other proteins, but also typical structures of globular proteins brought together. We could also take those complexes and fit them to the electron microscopy maps as we did for monomers, seeing that things just fit, like big complexes. Or these 2 proteins we could never before find in the map. People for decades were looking where they are in the complex, and here we just got a dimer and we could pinpoint the location. So now it's a long story, but our reactions were like that sometimes like that. And then what we decided to take this constructively and actually enjoy the process, and Agnieszka bravely trashed the entire year of work on homology models and fitting and building the model, and started over with a new modeling procedure, which is now kind of a standard. Taking the map, taking the AlphaFold models of proteins and subcomplexes. That time didn't use any crystal structures or no homology models, and then running assembly again to fit them to the map. And adding some new features to the procedure on top, like for instance, elastic network to maintain AlphaFold interfaces, or some other programs and approaches to model flexible linkers between domains, which were only now possible because of the high precision of the input models. And long story short, what it enabled us is to update the model that we had in 2016. So this is the model from 2016 that comprised 35 megadalton out of 120 megadalton, and the new model is 70 megadalton, which is around 90% of the structured, modelable kind of part of the complex. The other parts are disordered regions, mostly disordered regions that fill the channel of the complex. We learned many things. I don't have much time to really explain biology, and probably you are more interested in the computational aspects anyway, but it's just one example that this higher precision allowed us to place some proteins and understand how the nuclear complex is kind of glued together by flexible linkers and peptides going around the structure. The structure is also now used to actually understand that, how the transport through the nucleus works. So for instance, this is one of the first papers that takes the structure and grafts those disordered regions that form permeability barrier for the transport and runs molecular dynamics simulations to simulate how this barrier performs its function. But the revolution started. Now, if you talk among biologists, no one is surprised by AlphaFold anymore. It's already quite a time. And since then, AlphaFold, so first there was this hack, but in the meantime, DeepMind released a more designed and trained AlphaFold multimer version, which is trained on complexes and is way even better than what we had at the time. There are other programs, like academic programs from David Baker, RosettaFold, which is also a very good program for modeling and has also other features like soon modeling, post-translational modifications, ligands, and all kind of things. And some other implementations based on AlphaFold, like OpenFold, Unifold, and many others. So there's a huge flurry of explosion of programs, AI-based programs that were inspired by this transformer architecture designed by DeepMind. There are first programs for modeling using constraints actively. So first, what I showed was actually running AlphaFold and then doing again the classical approach of modeling with Monte Carlo. But there are now first approaches that actually try to use experimental restraints directly in the AI architecture, like AlphaLink. And also some programs now try to automate and also sometimes use AI to perform combinatorial assembly of bigger complexes out of these small components. Because AlphaFold still cannot model huge complexes because of the memory limitations. For instance, the last one, the AlphaFold multimer uses RL, uses reinforcement learning to perform these combinations. And these days, many, if not most, clarity microscopic structures are built using AlphaFold or RosettaFold as starting models. So you get a map, even if you get 3-angstrom resolution, you model AlphaFold of different pieces, fit them in, and then refine using molecular dynamics, for instance, into the map. Do I still have time? Yeah, I think so, 7 minutes. Okay, so we also developing tools around this revolution. So for today, I just selected one, which is Alpha Pulldown, which is a pipeline that we developed in the lab to facilitate protein-protein interaction screening with AlphaFold. And it has been inspired by or motivated by two reasons. So first, that when you have a complex like nuclear pore complex, you want to model different combinations. It's actually a lot of work to build all those combinations, score and analyze and so on. Second, you can imagine that if you can now predict models of protein interactions, maybe you can discover new interactions. So if you have one protein and then 100 other proteins, you can actually model all pairs to see which score well and predict new interactions. So we developed this package, which is a wrapper around AlphaFold. So it doesn't bring much algorithmic advances, but actually became very popular. So it can run in pull-down mode. So if you have one protein against a set of proteins to find those that interact, bind against praise. So it simulates an experimental pull-down experiment, like a purification. You can easily run different homologous modes. You can, like, if you have a protein and you know what is forming dimer, trimers, and so on, you can generate all of them very easily. You can calculate different subcomplexes of a complex, and also if you can take 15 proteins of your complex and run all against all to calculate the interaction network. And even though it's a simple wrapper, somehow it got very popular, and we think it's just because it's more user-friendly than the original version. It has some workflow speed improvements, so no algorithmic improvements. But it provides a graphical summary of the results and tabular summary with scores. It's open source, And how does it work? So for the kind of from the computational side, how the workflow is improved. So it's kind of splits the entire process into different stages. So one stage is the feature generation, which usually runs on CPU, and that's now parallelized. So you run it for all your sequences and create kind of database. And the inference, the structure prediction, runs on GPU, and then can reuse different parts or chunks of those features efficiently on GPU. And then also provides a set of analysis tools. And just finishing, so some of those opportunities that we have now in the integrated structure modeling and development, we put into opinion articles. So one published before the code was released and before even modeling complex was possible, put some predictions like that it should be possible, and it was. And there's another where we delineate opportunities in integrative structure modeling. So to acknowledge, I first would like to acknowledge some other researchers that work that kindly provided slides for the AlphaFold part of my presentation, and then people who did the work. So Agnieszka for modeling, Assembline was written originally by me, and then finished by Vasilios and Kai. Molecular Dynamics, which I didn't show, by Mark Siegel, and it's a long-standing collaboration within a kind of consortium or group of scientists working on the nuclear power complex with Martin Beck. Kind of leading the collaboration. And yeah, we also now try to apply AI to our computational modeling problems, and we have 2 postdoc positions for computer science postdocs open. So if you can contact me even if you didn't work in biology. And with that, thank you for your attention.